So we have looked at things that can control gene expression throughout the central dogma of biology, whether that is controlling the nature of chromatin so that it can be either expressed or not, whether that is controlling what's being transcribed, or whether that's the allosteric control of the protein itself in terms of the protein's shape. What other kinds of controls do we have in place? Certainly, certain kinds of cells express certain genes that others do not. For example, we have, again, we have the entire genome of the human body in every single cell. So here we have an example where liver cells will produce a blood protein called albumin, where cells in your eye that are literally in the lens of your eye will produce a protein called crystallin. The liver cell has no need for crystallin, okay, and the lens cell has no need for albumin. So the type of cell that they are determines what kinds of proteins that they'll produce. And that is done mainly by different activators. So here we have two genes, one in the liver cell that's going to produce this albumin, this blood protein, and it has activators that will allow the RNA polymerase to transcribe this DNA. It does not have the activators to transcribe the crystallin. Conversely, in the lens cell, there are no activators for the albumin blood protein, but there are activators that produce the crystallin. So two different cells producing two different proteins based on where those cells are at in the body. Now, there are other instances where we need several genes in the same pathway produced at the same time. And since eukaryotic cells are not as simple as the operon system in a prokaryotic cell, we will have things like transcription loops. Multiple chromosomes that can congregate in one spot. Genes can be transcribed from those individual the genes can be transcribed from those individual places on the DNA, producing all of the needed transcripts at once for whatever pathway is being produced. Now, the next thing we're going to take a look at is once we have something transcribed, how can we control what kind of proteins produced? And that comes down to the post-transcriptional control, comes down to RNA splicing. Okay? And if you'll remember, RNA splicing is not completely intuitive. Introns versus exons. Don't forget the exons are included in the DNA splice. The introns are not. Okay, so that seems a little bit backwards given their prefixes, but introns are not included and exons are expressed, is I believe where the X comes from in exons. Okay, so let's take a look at gene splicing, RNA splicing, and introns and exons in our next installment.